to easily detect it. So that's how you know that that glucose is different than normal glucose because of its radioactivity due to differing amounts
Mm.
of neutrons.
Okay. Thank you.
Mm-hmm. All right, so now we're going to talk about chemical bonds. These are the main chemical bonds that you have to know. You have covalent bonds, which is the sharing of electron pairs, ionic attraction, which is the attraction of opposite charges, hydrogen bonds, which is between a covalently bonded hydrogen atom and an electronegative atom, specifically nitrogen, oxygen, and fluorine. You have hydrophobic interactions between nonpolar and polar substances, and then van der Waals interactions, which is just an interaction of electrons between nonpolar substances. So now we're going to talk about water specifically. So water is extremely important in that for especially for hydrogen bonding. So since electrons are charged, the distribution of charges becomes polarized within the molecule. So if you see here, the oxygen is more electronegative than hydrogen. Because of that, oxygen pulls more electrons towards itself. Because of that reason, oxygen has a partial negative charge, while these two hydrogens have a partial positive charge. Next, over here, you see hydrogen bonding. Now, what you have to remember is that hydrogen bonding forms between molecules, not within the molecule itself. So as you see here, the bond between the hydrogen and oxygen within the molecule is a covalent bond. However, the bond between this molecule and that molecule is a hydrogen bond specifically. So hydrogen bond often forms between molecules. Next, why are hydrogen bonds important? So they help stabilize adjacent molecules to make arrays or crystals, as you see here. And they're especially important in order to stabilize 3D configurations of especially large molecules, such as proteins. So here, now we're going to talk about ionic bonds. So ionic bonds are exceptionally strong because they involve the complete transfer of electrons between atoms, which allows for a strong electrostatic interaction. As you see here, sodium has an unfilled quartet. Chlorine also has an unfilled ring. So chlorine takes the electron from sodium. It steals it from sodium. Now sodium has a completely filled ring. Chlorine has a completely filled ring. So now the atoms are both extremely stable and have full electron shells. Now, interestingly here, so as I said before, ionic bonds hold sodium and calcium together. So when sodium and calcium is dissolved in water, they both attract different parts of water. So because sodium, as you see here, is a positive ion, it attracts the negative, so it attracts the water molecules, but it attracts the negative oxygen atom to it. So the negative oxygen atom is facing the sodium ion, but chloride is a negative ion, so chloride attracts the positive hydrogen ions towards it. So even within the water, even for water, it can attach the different ions differently based on if the ion is positive or negative. So lipids are exceptionally important for hydrophobic interactions. So as you see here, lipids have a hydrophilic head group, which can freely interact with water, and they also have a hydrophobic alkyl group. So when they come into contact with water, the water molecules become highly ordered and form cages. The reason why they form cages, as seen here, is in order to maintain their hydrogen bonds, because they can't interact with the hydrophobic alkyl group. So they form this cage so that the hydrogen bonds, as you can see here, can be maintained. However, this dispersion can lessen as you increase the amount of lipid molecules. So as you see in your right, when you have three lipid molecules next to each other, only the lipid portions at the edge force the ordering of water. So fewer water molecules are ordered, entropy is increased, which allows reactions to occur. And then micelles, which is as you see in phospholipid bilayers, which we'll talk about later, all the hydrophobic groups are separated from water, so they're not interacting with water at all. Because of that, any ordering of the water into a cage is minimized and entropy is increased to the max. So cellular environments and tonicity, I believe you did a lab about it. This is simple. When a cell is in an isotonic solution, there's no net water movement, which means the extracellular solutes and intracellular solutes are even. In a hypertonic solution, there's more solutes on the outside of the cell than inside of the cell. Because of that, water moves out of the cell and the cell shrinks. In a hypotonic solution, there's more intracellular solutes than extracellular solutes. Because of that, water moves into the cell can swell and then eventually burst. So now
I
we're
have
going to talk.
another question here. Um,
Okay, yeah.
so when you say that it eventually burst, does it really mean the cell dies off?
Yeah, so what happens is that if there's an extreme amount of solutes within the cell, water is
Mm-hmm.
going to keep on rushing into the cell. The cell obviously has a cell membrane. It's not going to be able to hold all of
This would be
the.
an osmosis process, correct?
Yeah. So the cell's obviously not going to be able to hold all of that water. So water rushes in. Eventually, the cell membrane breaks and the cell gets lysed.
And in the opposite, when it shrinks, does it die also,
Yeah,
or
it
it's
dies
almost
also because the
okay?
reason it dies, it doesn't have any water or nutrients to be able to maintain any of the intracellular
Hmm.
organelles.
Got you. Thank you.
Mm-hmm. All right, so now we're going to talk about buffers. Oh, actually, sorry. First, we're going to talk about weak acids and bases. So in weak acids and bases, we use the Bronsted-Lowry definition, which is proton donors and proton acceptors. So here, as you see, weak acids partially break apart. So acetic acid or acetate, they don't release all of their hydrogen ions. So they only partially break apart when they interact with other molecules. Strong acids like HCl, they completely break apart into its hydrogen ions and then chlorine ions, or H2SO4. They completely break off into its individual parts. Same thing for weak bases. They partially break apart, and then strong bases, they completely break apart. So you're going to have a lot of sodium ions and a lot of OH minus molecules. So what are buffers specifically? So buffers are weak acids and they have an ability to ionize and they're reversible. So weak acids are better as buffers than strong acids because they only partially break apart. They can maintain an equilibrium that can neutralize any strong bases that are added or any strong acids that are added. So as you see here, bicarbonate is one of the most common buffers in our body. So bicarbonate partially breaks apart into HCO3 minus and hydrogen ions. Sorry, someone raised their hand.
Yes. So, for ionizing with the H plus, the
Yeah.
word ionize, does it mean that it's gonna accept the proton from another atom?
Yeah. So basically what happens here, if a strong acid breaks apart, whatever it's reacting with will be able to take all of the hydrogen ions from it. But in a weak acid,
Okay.
as you see here, H2CO3, it only breaks apart into HCO3 minus. So it still manages to keep some of the hydrogen. So it doesn't fully break apart into its individual components. It only partially breaks apart.
Yeah,
So
I
the
got
free,
that. I
yeah,
got that. So for breaking it down, it's like the hydrolysis.
yeah, it's hydrolysis.
Yeah, hydrolysis process. Is that what you call, or like there is another name for it?
No, that's what you call it.
And for adding it, that would be dehydration.
Yeah.
What's the word ionize has to do? I'm a little confused with that.
So ionize means we're breaking it apart into its ions. So here you have HCl, for example. HCl isn't an ion in and of itself. However, because it's a strong acid, if you react it with something, it breaks apart into its individual ions. So it breaks apart into hydrogen protons and chlorine anions, Cl minus and H plus. So that's what
So it's
ionization means.
mostly because of the positive and negative charges that those atoms has.
It's because of how electronegative the atoms are.
Okay,
So
gotcha.
chlorine is extremely electronegative. It'll break apart with hydrogen individually because it's a strong acid.
Hmm, thank you.
Mhm. So, like I mentioned with buffers, as you see here, when H2CO3 reacts with NaOH, it still keeps some of its hydrogen ions. So any free-floating hydrogen concentration remains unchanged, which allows it to actually make sure that there isn't significant changes in pH. So as you see here, without any pH added, if a base is added to an acid without any buffers, it immediately decreases significantly. However, in the presence of a buffer, any addition results in relatively small changes. So this red dot here is the midpoint where the buffer pH is equal to the pKa. So it's the point at which half of the weak acid is titrated with weak base. Usually the buffering range is around plus or minus 1 pH unit above the pKa. So here the pKa is 5. So the buffering range is between 4 to 6. pH is negative log H plus. And then what you have to remember about the buffering range is that it's not a infinite capacity. The buffer itself has a range where if any added base goes above that range, if the capacity is exceeded, any added base will greatly increase the pH. So the buffer has a limited quantity of how much it's able to affect the pH by, which is usually plus or minus 1 above the pKa. So what are isomers? So isomers are molecules with the same chemical formula, but their atoms are arranged differently. So structural isomers, as you see here with butane and isobutane, their atoms are joined differently from each other. So as a result, they have different chemical properties. Then you also have cis and trans isomers. Cis isomers means that they're on the same side of the double bond. Trans isomers mean that the molecules attached to the double bond are on opposite sides. So once again, cis, same side. Trans is opposite side. Hello? Oh, sorry, I saw another hand.
Yes. So is this only because of their structure? They work in different ways because butane works as a fuel and isobutane works as a something with the refrigerator, correct?
Yep. So purely because of the structure, it changes their chemical properties exceptionally. So that's the main thing about
Okay.
structural isomers. Different arrangements can cause different chemical properties.
Okay. Thank you.
So, and then cis, once again, cis is same side, trans is opposite side of the double bond. And then you have optical isomers. Optical isomers are mirror images of each other. And then optical isomers can only occur when there are 4 different bound groups to one carbon. So as you see here, you have the carbon in the middle, and you have 4 completely different groups. So once you have 4 completely different groups, you can have optical isomers, which are mirror images of each other. If any of these 2 groups are the same, then they can't form a mirror image of each other. So now we're going to move on to chapter 3 and 4, which is macromolecules. So, all right. So one of the most important motifs in biology is structure is equal to function. As shown with the isomers, any structural isomers, if there's a change in structure, there's a change in function. And that's also true for molecules as well. So the basics of any macromolecules, you have your individual subunits. Individual subunits combine through covalent bonds to form macromolecules. Then these macromolecules attach to other macromolecules to form macromolecular assemblies. So for example, amino acids can form proteins, proteins can combine and form ribosomes. So you have what are known as monomers and polymers. So monomers are the units that are the building blocks of macromolecular polymers. So here the monomers are a specific sugar, such as glucose, amino acid, or nucleotides. And then they combine with other amino acids, nucleotides, sugars, etc., to form macromolecules, such as polysaccharides, proteins, and nucleic acids. So condensation is a way that polymers are synthesized from monomers. So what happens during condensation? Water is removed. So here you have an OH, here you have a hydrogen. The oxygen attaches to the hydrogen, and because oxygen already has 2 hydrogens attached to it, it breaks off from this molecule over here. Then it's able to combine to this monomer over here. So a covalent bond is formed. And then this can continue infinitely to form a polymer. So this is known as condensation, and it's also sometimes known as a dehydration reaction. Then you have hydrolysis. Hydrolysis is how you break down polymers into monomers. So polymers are breaking down, water molecules are added to a polymer. When water molecules are added to a polymer, then one monomer attaches to an OH atom, another monomer attaches to a hydrogen atom, and then the covalent bond is broken down. So now we have lipids. So lipids are extremely important. They're part of our phospholipid bilayer. They help make sure that random molecules aren't able to go through. So triglycerides, as you see here, are synthesized from glycerol through condensation reactions. So this is just another example of how condensation reactions work. You have the monomers here, and then you have 3 fatty acid molecules. Through condensation reactions, you're able to attach it to the glycerol to form triglyceride. So now we're going to talk about phospholipids themselves. So they're the building blocks of biological membranes. They contain a hydrophilic head, which is able to interact with the environment outside the cell or inside the cell. They also contain a hydrophobic tail, which actually forms the bilayer. So usually contains two fatty acid chains connected through a glycerol as well as a phosphate. And it's also amphipathic, which means it has a polar region and a non-polar region. So it has both. So once again, polar head, non-polar tail. And this is the phospholipid bilayer, so which is a micelle, as we mentioned before. It has hydrophilic heads, which is able to interact with the environment outside the cell and inside the cell, and then the hydrophobic tails in the middle. And then van der Waals interactions help strengthen these hydrophobic interactions. So now sugars, which make up carbohydrates. So monosaccharides are often referred to as sugars, and one of the most common form of sugar is glucose. And then glucose can combine with other sugars such as lactose, galactose, etc., to form cellulose, starch, and glycogen, which are all extremely important and have several functions within our body. So polysaccharides can be modified. Once they're modified, they have special functions. So as you see here, this polysaccharide has phosphate groups attached to it. When phosphate groups are attached to it, this helps work in gaining energy from glucose through ATP. Another is amino groups can be added to polysaccharides. When amino groups are added to polysaccharides, as you see here in galactosamine, it's an important component of cartilage. So there are many ways where you can interact with a polysaccharide and change its function. Another one, for example, is chitin, where you add a N-acetyl group. So N-acetyl groups allows for more hydrogen bonding, and that allows for a strengthened molecule. And now, why is this important? Chitin is commonly found as an exoskeleton of insects. Because of that, the N-acetyl group helps create these hydrogen bonds, strengthens it, which makes the exoskeleton of insects stronger as well. So now polypeptides. Polypeptides have an amino acid, which is an aminated carboxylic acid. They have both an acid and a base, so acid here and the base here. And then the carboxyl group is an acid or a proton donor. Amino group is a base proton acceptor. And then these amino acids can eventually form polypeptides. So polypeptides consist of repeated peptide bonds between the amino group and the carboxyl group. So the amino group and the carboxyl group, once again, condensation reaction, water comes out, allows for a bond to form between the carbon and the nitrogen, and then this proceeds to form polypeptides. And polypeptides grow from the amino group to the carboxyl direction. So if there's a question on what direction do amino acids grow, they grow from the amino group to the carboxyl direction. So monomers are added at the carboxyl terminus. So cysteine or disulfide bridges can be formed between cysteines within the same polypeptide. So cysteine is a specific amino acid, which is known for having a sulfur attached to it. So cysteine molecules can form within the same polypeptide chain, can form disulfide covalent bonds where hydrogen that's attached to the sulfur can leave, allowing the sulfur to interact with each other. This creates a stronger bond and a stronger molecule. So as you see here, once again, you have polypeptides. So the primary, so we're going to talk about primary, secondary, tertiary, quaternary structures. So the primary structure defines the protein identity. So it defines what type of protein it is, whether it's insulin, albumin, hemoglobin, etc. They all differ in their amino acid sequences. So proteins of the same type have similar amino acid sequences across species. So for example, insulin has similar sequences across all mammals. So any modifications can have dramatic functional consequences where either it doesn't work at all or it may mutate, which obviously is not good. So now you have secondary structures. Within secondary structures, there are two types of structures that can be created. You have an alpha helix and a beta pleated sheet. So alpha helixes allow hydrogen bonds to be formed between the amino and carboxyl groups, which helps stabilize the polypeptide backbone in a helical structure. Alpha helixes are common, so side chains project away from the core of the helix. So any side chains of the amino acid, they project away from each other. But secondary beta-pleated sheets, hydrogen bonds also form between the carboxyl and amino groups, but this helps stabilize multiple chains into sheets. And then the uh, R chains are sometimes projecting away from the core, sometimes projecting inside the core. So depending on the structure, depending on the primary structure of the amino acid sequence, it can either turn into an alpha helix or the beta-pleated sheet. It depends completely on the structure, the primary structure of the amino acid. Next, you have a tertiary structure. So tertiary structure forms when R group interactions start to fold the polypeptide within each other. So now beta-pleated sheets can interact with alpha helixes. Alpha helixes can interact with other alpha helixes. And then cysteine disulfide bridges, as mentioned before, help stabilize these tertiary structures. And quaternary structures are just multiple tertiary structures bound to each other. So this can just be one subunit of multiple. So hydrophobicity can change polypeptide configurations. So polypeptides that have predominantly hydrophilic side chains remain in the cytoplasm as it's a hydrophilic region. So they usually assume a linear configuration. Polypeptides that have hydrophobic side chains tend to seek out hydrophobic environments. So which is lipophilic, they like fatty acids. So they tend to be embedded within the cell membrane among the fatty acid tails. What if they have both regions? So polypeptides that have both hydrophobic and hydrophilic regions tend to fold into itself with spherical structures, and the structure is stabilized by any hydrophobic interactions. So as you see here, you have an unfolded polypeptide that has polar side chains and non-polar side chains. It's able to fold into itself and create a sphere. So you have a hydrophobic core in the middle, and then you have the polar side chains on the outside, which allows it to bind to any aqueous environments. So these are all the different proteins that can be found in our body. I would say definitely memorize each of the proteins and their basic functions rather than like what specific enzymes. For now, definitely memorize what the categories are and their functions as well. Next, we're going to talk about polynucleotides. One of the most common polynucleotides is DNA and RNA. So polynucleotides are made from a base. You have your pyrimidines, which is cytosine, thymine, and uracil, and your purines, which are adenine and guanine. So each of these bases bind to a sugar, which is ribose or deoxyribose, and then that forms a nucleoside. Once that nucleoside attaches to a phosphate, it forms a nucleotide. So deoxyribose, and then one deoxyribose nucleic acid is DNA, ribonucleic acid or ribose nucleic acid is RNA. So RNA versus DNA. So first in DNA, DNA adenine binds to thymine and it has a double bond. Guanine binds to cytosine and it has a triple bond. So that could also be a question on the exam. Which pairs have a triple bond? Which pairs have a double bond? In RNA, adenine binds with uracil instead of thymine, while guanine binds with cytosine. Furthermore, RNA is single-stranded, DNA is double-stranded. Now, how are nucleotides attached to these RNA or DNA to elongate it? So any nucleotides are attached to the 3' end of the polypeptide chain. So elongation occurs from the 5' end to the 3' end. So 5' end to the 3' end. So any new nucleotides are going to be added here and here. For RNA, any new nucleotides are going to be added here. So once again, distinguishing RNA from DNA, RNA has ribose, DNA has deoxyribose. It does not contain an oxygen. RNA is adenine, cytosine, guanine, and uracil, while DNA is adenine, cytosine, guanine, and thymine. And then RNA is usually single-stranded. Most of the times, it's very rare where RNA is double-stranded, while DNA is almost always double-stranded. Another important rule that you have to memorize, because there may be a question on the exam, is Chargaff's rule. Chargaff's rule is that the amount of adenine is equivalent to the amount of thymine. Oh, sorry. Are the double bond and triple bond formations the same for RNA? Yeah, yes, it is. So for Chargaff's rule, whatever percentage of adenine you have is the percentage of thymine you have. Similarly, any percentage of guanine you have is the percentage of cytosine you have. So there could be a question on the exam. If you have 20% adenine in your DNA molecule, how much cytosine do you have? Well, if you have 20% adenine, you have 20% thymine, which is 40% total. That means cytosine and guanine make up 60% of the remaining DNA. So because cytosine is equivalent to the amount of guanine, cytosine is 30%, guanine is 30%. So that's how you figure it out. So that's Chargaff's rule. A is equivalent to T, G is equivalent to C. And for RNA, A is equivalent to U. So now we're going to talk about chapter 5, the cell. Yeah, the ratios are 1-to-1 for adenine and thymine and guanine and cytosine. The ratios aren't 1-to-1, for example, for adenine and guanine because they're not bonded to each other. Specifically, the pairs are 1-to-1. Yeah, of course. All right, so chapter 5, the cell. So the importance of surface area to volume ratio. So volume determines the amount of chemical activity that occurs in the cell per unit time, while surface area determines the amount of substance that's able to pass through the cell boundary per unit time. So the greater the surface area to volume ratio, the more efficient the cell is at material exchange. So here, the smaller cell has a greater surface area to volume ratio, which allows it to be more efficient. It's the same reason why we're going to talk about later. The mitochondria has cristae, which are folds to increase that surface area to volume ratio. Similarly, our brain has grooves and ridges in order to increase that surface area to volume ratio. So now you have prokaryotes. Prokaryotes include all bacteria, all archaea. So prokaryotes are unique as they are unicellular organisms that don't contain any membrane-bound organelles. So capsule is a protein coat that surrounds it. You have a cytoplasm, ribosomes. The nucleoid, prokaryotes don't have a true nucleus, so they have a nucleoid, which is a small region that contains a circular DNA strand. It doesn't contain a full chromosome. You have a plasma membrane, and you have a flagellum, which allows it to move. I would recommend knowing where the structures are relative to each other and memorize their basic functions. Because sometimes there's a 4-question part where you're given a photo of one of and you're told to identify any 4 or any 2, 3 parts of the cell. So I would definitely recommend knowing their function and where they are relative to each other. So now you have eukaryotes, specifically animal cells. So eukaryotes, they do contain membrane-bound organelles. They can be multicellular, and they also have several important parts. So one example is the nucleolus. The nucleolus is responsible for creating ribosomes. They have a true nucleus, which contains all the DNA within the cell. You have the cytoskeleton, which maintains the structure, peroxisomes, which breaks down lipids, etc. So once again, memorize every main organelle within the eukaryotic cell. You also have plant cells, which are also eukaryotes. Important parts of the plant cell to memorize: the vacuole. The animal cells have vacuoles as well, but definitely not as pronounced as in plant cells. Vacuoles in plant cells contain water, contain nutrients. You have plasmodesmata, which allows for nutrients to be able to be transported across multiple plant cells. So once again, memorize all of this. So membrane cycling. What is membrane cycling? So membrane cycling, first you have lysosomes, which originate from the Golgi apparatus. So digestive enzymes are concentrated and packaged into what are known as lysosomes. So these lysosomes have digestive enzymes within it, and they're pinched off and they become primary lysosomes. Then any food particles from the outside of the cell are taken into the cell through phagocytosis. Now you have a phagosome, which is a vesicle that contains these food particles. The primary lysosome and phagosome combine to create a secondary lysosome. Eventually, any molecules that are digested and that are molecules that are generated by the digestion from the secondary lysosome diffuse into the cytoplasm. Any undigested molecules are then released out of the cell. And then lysosomes are also able to digest any intracellular organelles and debris that's on the inside, and that's known as autophagy. So now we're going to talk about the cytoskeleton. Cytoskeleton has several functions. It maintains and supports cell shape, provides support for cell motility or cell movement, and it also provides scaffolding for intracellular transport. So here you have microfilaments, which are made up of actin, and they interact with several strands of other proteins, specifically myosin mainly, which is a motor protein that moves along it. You have intermediate which are made up of extremely fibrous, tough proteins. They're able to combine to create a rope-like assemblage. Intermediate filaments help make sure the cell structure is stabilized and the shape is maintained through any stress. Then you also have microtubules. Microtubules are hollow cylinders made up of tubulin. There's alpha and beta tubulin, and motor proteins such as kinesin and dynein move along the microtubule tracks. The flagella of prokaryotes and in our sperm as well is made up of microtubules. So kinesin transport. So how does kinesin move along the microtubule tracks? It basically walks similarly to a human. So you have the kinesin motor protein here attached to a microtubule. It's cross-linked to this vesicle here. Then one part of the quote-unquote feet is unbound and then moves forward. Then the previous foot is unbound and then also moves forward. So any detachment and reattachment of the kinesin causes it to walk along the microtubule. And it always moves particles from the minus end to the plus end of the microtubule. So now you have the mitochondria. As everyone says, the powerhouse of the cell. The mitochondria is extremely important as it converts potential fuel molecules into ATP. It has an outer membrane as well as an inner membrane. Within these two membranes is the inner membrane space. Within the inner membrane itself is the mitochondrial matrix. And then the inner membrane protrudes into the matrix in order to form cristae. Once again, the importance of cristae in creating these folds is to increase the surface area. If you have an increased surface area, you're allowing better transmission of molecules and better efficiency of converting fuels into ATP, especially for the mitochondria. It's exceptionally important. You have chloroplasts, which are found in plant cells, which are the site of photosynthesis. They help convert light energy into chemical energy. They also contain two lipid bilayers, inner membrane and outer membrane. So the inner membrane surrounds the stroma here. So the stroma is responsible for structural support and nutrient and gas exchange. Then it also contains the thylakoids. The thylakoid is responsible for the light-dependent reactions of photosynthesis, which you'll learn about in a future chapter. And it also has the granum. The granum helps increase surface area to make sure the light-dependent reactions are able to move efficiently. So here you have the endosymbiotic theory. The endosymbiotic theory states that several intracellular organelles, particularly the mitochondria and chloroplasts, were once independent, free-living prokaryotic cells. Eventually, they were absorbed within larger cells, and once they were absorbed, they were able to live symbiotically within that cell. So the reason that people believe the endosymbiotic theory is true is that both mitochondria and chloroplasts are able to self-replicate. So these are very unique organelles that are able to self-replicate. They have their own DNA and ribosomes that are unique to them, and they have genetic and biochemical characteristics that are similar to prokaryotes rather than eukaryotes. So we'll post the slides later. This is a 7-minute video just on how the cell works. Just to save time. If we have time at the end, I'll play it, but I'll move on to chapter 6 now. So chapter 6, biological membranes. So as we mentioned before, biological membranes are made of lipids. So you have saturated and unsaturated fatty acids. Within saturated fatty acids, the acyl chain here has no double bonds. But in unsaturated fatty acids, they do have double bonds. As you see here, there's 2 double bonds. So what's the importance of these double bonds? When double bonds are formed, they create a kink. If a kink is formed, these fatty acids are not able to be packaged as tightly amongst each other. As you see here, it's packaged for saturated fatty acids, they're packaged really tightly. But for unsaturated fatty acids, they're not able to be packaged as tightly amongst each other. Because of that, saturated fatty acids tend to be solid at room temperature, specifically like animal fats, for example, while plant oils tend to be liquid at room temperature because they're not packaged as closely to one another. So this may cause a heart attack. What is this? This is cholesterol. So cholesterol is extremely important. It's found in our membrane. So it changes the membrane fluidity depending on the fatty acid composition of the other lipids found in the membrane. So if you have a lot of cholesterol, eventually this will be able to thicken. So because cholesterol can change the fluidity of the membrane itself, it can make the membrane fluidity almost zero, creating plaques, making sure no molecules are able to go through. If that happens, if you have lots of plaques, that can eventually cause a heart attack. and fluidity also decreases with declining temperature. So this is what's known as the fluid mosaic model, which is that the membrane is not just lipids. There are several parts to our membrane. Obviously, you have the phospholipid bilayer, but then you have carbohydrates, which help control the membrane fluidity. You also have peripheral membrane proteins, which don't penetrate the bilayer, but they're next to it. You have integral membrane proteins, which cross the entire phospholipid bilayer. Some penetrate only partially. So the fluid mosaic model, you have lots of moving parts. Proteins, the phospholipid tails, cholesterol, etc. They all work together to help control what materials are able to get through or not. So what is able to pass through? So in order for it to pass through, it obviously has to go through the nonpolar region of the fatty acid tails. So hydrophobic molecules are able to pass through extremely easily. For example, carbon dioxide, oxygen, nitrogen, or steroids. Polar molecules, only very small polar molecules are able to get through. Once again, because they have to interact with the non-polar region, they can't be large. They have to be extremely small, so water, glycerol, or ethanol is able to go through, but at a much reduced rate compared to hydrophobic molecules. And then large polar molecules is not able to get through. Charged molecules as well are not able to get through. Charged ions themselves are specifically only pumped through. So which we'll talk about now. So here you have passive membrane transport. So membrane channels themselves are integral, which means they're part of the actual membrane itself. So they're integral membrane proteins. The passageway of the channel has hydrophilic amino acids, and then hydrophobic amino acids face the outside of the channel towards the fatty acid tails. So once again, the passageway of the channel has hydrophilic amino acids. Hydrophobic amino acids face the outside of the channel towards the fatty acid tails. So ions are able to passively transport through the cell. Usually a stimulus has to bind to these channels. So what happens? They're often gated. So molecular signals, pH, physical pressure, or any voltage changes can all be a stimulus molecule. This binds to the transmembrane protein. Once that happens, the ion channel is open or closed. In this case, the stimulus molecule binds to it, it's open. Once it's open, the ions are able to passively go through the molecule. With ion passive transport, though, they are specific for only one type of ion. So, for example, if chloride ions are getting through, sodium ions aren't able to get through. It's specific for only one type of ion. Next, you have what are known as carrier proteins. So carrier proteins is also part of facilitated diffusion. So carrier proteins involves binding to the actual substance itself and then allowing it to go through the membrane into the inside of the cell. And with carrier proteins, that actually allows diffusion in both directions. So glucose from the outside of the cell is able to bind to the carrier protein, go to the inside of the cell if there's a low glucose concentration here in order to maintain equilibrium. But if there's a high glucose concentration on the inside of the cell, it's able to go the opposite direction as well. So carrier proteins are bidirectional. Now we're going to talk about active transport. For active transport, it requires energy, usually ATP, and then ions are moved against their concentration gradient. So you have three main active transport proteins. You have a uniporter, which only transports one substance in one direction. Yeah, carrier proteins are a form of facilitated diffusion. So uniporters transform ions specifically in one direction. Symporters can transport two different substances in the same direction. So for example, there are sodium glucose symporters. So sodium and glucose can go from the outside of the cell to the inside of the cell. And then there are antiporters. Antiporters are extremely important because they transport two different substances in opposite directions. Sodium is able to be brought in, potassium is able to be brought out. So primary active transport. So here we're going to talk about the ATP being used as a source of energy, particularly the sodium-potassium pump, which, as I mentioned previously, is a antiporter. So here, a concentration gradient is established. So eventually, what you have here, ATP binds to the sodium-potassium pump. Once ATP binds to the sodium-potassium pump, hydrolysis of ATP occurs, where the protein is completely changed. or the protein shape is changed. Once the protein shape is changed, sodium is released to the outside of the cell. If sodium is released to the, sorry, I saw a chat. Oh, sorry. ATP binds to the sodium-potassium pump. Once ATP binds to the sodium-potassium pump, the ATP gets hydrolyzed, so phosphate binds to the sodium-potassium pump. Because of that, sodium is able to be taken outside of the cell, potassium is able to be brought into the cell. And then eventually phosphate gets released, which allows the sodium-potassium pump to be returned to its original shape. And once again, the cycle repeats eventually. So the sodium-potassium pump is extremely important for our neurons whenever action potentials occur. All right, give me 1 second. So now we're going to talk about endocytosis, which is how cells obtain nutrients. So penocytosis is specifically taking in fluids and solutes from the outside, while phagocytosis is taking in larger substances such as bacteria. So what happens is what's known as membrane invagination, where the molecules are internalized, the plasma membrane starts to fold inwards within each other, it engulfs the material and then forms a vesicle, which allows it to move it into the body. So penocytosis also creates this invagination vesicle. However, it's much smaller than phagocytosis, obviously. And then what you see at the bottom here is receptor-mediated endocytosis. So receptor-mediated endocytosis is highly specific. It depends on receptor proteins, which are integral membrane proteins. Once again, they're within the transmembrane. So they bind to specific substances. Once they bind to the specific substances at these sites of internalization known as coated pits, these coated pits then start to move inwards to each other. Once again, similarly as penocytosis and phagocytosis, it moves inwards towards each other, creating a vesicle. And then the vesicle itself is coated with clathrin, which allows the vesicle to be protected as it moves into the cell. So exocytosis is essentially, once again, the opposite. You're moving material outside of the cell. So a secretory vesicle binds to the membrane. Any indigestible substances are within the secretory vesicle. It binds to the membrane, and then eventually the vesicle releases all of its materials outside of the cell. So it's not just indigestible substances. Sometimes it's also hormones, neurotransmitters, digestive enzymes that need to be transported to a different cell. So cell junctions, how do cells communicate with each other? So cells form junctions among themselves in a tissue, and there are three main types: tight junctions, desmosomes, and gap junctions. So tight junctions are found in epithelial cells. They, oh, sorry. They tightly link the two cells together. Now, tightly linking the two cells together makes sure there's no movement of substances between the cells. So with tight junctions, the main function is it prevents any substances from being transported from one cell to another. So it restricts any migration of proteins, phospholipids, enzymes, etc. Desmosomes are a welding in a way. So they weld the two adjacent cells together, and then it helps promote cell interactions because of the adhesion. And importantly for desmosomes, they help make sure the cell doesn't break apart during any stretching, when their muscles working, etc. It helps make sure that the cells don't break apart. Gap junctions are typically found in cardiac cells. The main importance of gap junctions is they want to promote communication between the cells. So they're made up of protein channels called connexons. These connexons span the plasma membrane of two adjacent cells and protrude from them slightly into the cytoplasm. And then connexons are made up of connexins, which snap together to generate a pore. So it helps make sure that substances are able to go through from one molecule to another. So now we're on our last chapter. I'm gonna drink a little bit of water. Give me a second. So chapter 7, cell signaling and communication. So the general pathway for any signals is the signal transduction pathway. A signal molecule binds to a receptor. That receptor causes changes within the cell. For example, here, a signal transduction molecule becomes activated. Now, this signal transduction molecule can cause both short-term changes, where it activates other enzymes or moves the cell. It can also create long-term changes, where DNA transcription can be altered as well. Oh yeah, and these are known as responders. So you have a signal here, you have your receptor which binds to the signal, and then the receptor activates your responders. So there are several methods of cellular communication. There's autocrine, juxtacrine, paracrine, and exocrine. So autocrine is when any signal that's sent out affects the signaling cell itself. So as you see here, a signal is sent out, it affects a different part of the cell. So something could be sent from the Golgi apparatus, and then from the Golgi apparatus, it goes and affects the mitochondria, for example. Then you also have juxtacrine. Juxtacrine is when you affect adjacent cells, so cells right next to each other. So here, the signal is released, affects the adjacent cell here. You also have paracrine. Paracrine is when you affect neighboring cells, so the signal is sent out, affects the target cell somewhere else. And for these signals, there needs to be specific receptors. If there aren't any specific receptors, the signal isn't able to be bound to it. You also have exocrine here. So the importance of exocrine delivers cells outside of the body. So here, the secreting cell sends out signals. It goes through the bloodstream, affects the target cell elsewhere. Or here, they send the signal, affects them here. So it delivers it outside of the body. Now you have agonists and antagonists. So with agonists and antagonists, they're extremely important because binding of a ligand can change the configuration and affect the receptor complex. So with agonists specifically, they mimic whatever signal. So they mimic the ligand that binds to the receptor. So agonists help promote whatever signal it is. Antagonists, however, prevent binding of the natural ligand. So caffeine is one of the most popular antagonists. Typically what happens, adenosine binds to dopamine receptors, resulting in less dopamine being transmitted throughout the body. However, caffeine is an antagonist, which means it prevents the binding of adenosine. If caffeine binds to the receptor that adenosine can't bind to, dopamine levels will never go down. Dopamine levels remain high, which is why caffeine works so effectively. So phosphorylation is an extremely common transduction mechanism. So what happens is that you have a target protein. Sorry, you have a target protein and ATP. A protein kinase then comes in, which then allows the target protein to be bound to a phosphate. When the target protein is bound to a phosphate, from the ADP, it creates an altered shape and function. So here is a protein kinase receptor. So what you see here is insulin. Insulin binds to the insulin receptor. Once insulin binds to the insulin receptor here, the receptor kinase is able to autophosphorylate itself. So the phosphate group goes to any response substrate. The phosphate group attaches to the response substrate, which allows it to become activated, which causes cellular responses within the cell. So it can work both ways. It can either activate or deactivate a protein. So phosphorylation works both ways. So here, as you see here, ATP becomes ADP. The phosphate attaches to any phosphorylated protein. If it activates it, then the response signal increases. If it deactivates it, it makes sure there is no response signal. Eventually, so how do you get rid of that phosphate though? You have an enzyme known as phosphatase. Phosphatase gets rid of the phosphate on that protein, so that protein becomes dephosphorylated. It returns to its original state, which was either active or inactive. It depends on the protein itself. Next, you have G protein coupled receptors. G protein coupled receptors are extremely important for multitude of reasons. So the pathways are similar for almost every sort of receptor. So here, the outside of the cell, a signal binds to the G protein linked receptor. Next, you have an inactive G protein. Because that signal bound to that G protein linked receptor, that inactive G protein becomes activated. That activated G protein then goes and binds to this effector protein. That effector protein becomes activated, which then causes further signals. So let's talk about one example, olfaction, which is our ability to smell. So the ligand, which is the molecule that's binding to the receptor, is any odorant that you smell. The receptor is the G protein linked receptor here. Odorant binds to the receptor. Then the G protein becomes activated. Adenylyl cyclase, which is your effector protein, was inactivated, but then the GTP binds to adenylyl cyclase. When it binds to adenylyl cyclase, that effector protein causes a response. What is that response? It turns ATP into CAMP, which is a secondary messenger, which we'll talk about later as well. So it turns ATP into CAMP. CAMP has a secondary message. What's its message? It needs to open this channel. It binds to this channel, which opens it, allowing sodium and calcium to go inside, which this is a symporter. So pathogens can interfere with G protein functions. So one way, for example, the cholera bacteria. What happens with the cholera bacteria? It right here, it binds to the G protein. Once it binds to the G protein, it prevents any hydrolysis of GTP. If GTP can't lose its phosphate, it's persistently activated. So the switch is turned on permanently. The switch cannot be turned off. So now this effector protein is activated permanently, which can cause, obviously, if you have a repeated signal occurring several times in a row, that can cause severe damage to the cell. So here it's causing the signal to be permanent, but there's also an opposite modification as well for other bacteria. The whooping cough bacteria can bind to the G protein here. When it binds to the G protein here, it prevents any interaction with this receptor. Because of its prevention, it's permanently inactivated. No matter how much this receptor is activated by the signal, it can't turn on the G protein due to the toxin binding to it. So now we're going to talk about secondary messengers. So secondary messengers are low molecular intra or intercellular signaling molecules. Some common examples are CAMP, cGMP, DAG, IP3, calcium, and nitric oxide. So they're also part of the signal transduction cascade. The importance of secondary messengers, they alter the activity of enzymes or other proteins. As mentioned here, CAMP, which is a secondary messenger, it changes the activity of this protein. So in this case, it activates it, but it can also deactivate it, etc., depending on other proteins. And they help participate in the signal amplification process. So nitric oxide is another common example for a gaseous secondary messenger. Acetylcholine binds to this G protein linked receptor. This G protein linked receptor then activates IP3 and DAG transduction systems in endothelial cells. So what they do, they attach, IP3 attaches to this calcium transmembrane protein. Now calcium is able to go inside the cell. Once calcium is able to go inside the cell, it's able to make sure that nitric oxide synthase is turned on. Nitric oxide synthase turns on. Now you have lots of nitric oxide. What this does is that it diffuses into your smooth muscle cells, promotes the production of cGMP. cGMP is another secondary messenger, and cGMP then relaxes any smooth muscles. So signaling cascade and signal amplification. So this is an example of how secondary messengers and G protein linked receptors, all of these receptors all work together to create a signal transduction pathway. Epinephrine binds to this receptor. This G protein linked receptor becomes activated. It then activates adenylyl cyclase. Adenylyl cyclase then converts ATP to CAMP, which is a secondary messenger. Now, CAMP turns glycogen synthase, which produces glycogen. It makes glycogen synthase inactive. So now glycogen will not be formed because you want to break it down. And CAMP also has a secondary function. It turns protein kinase A active. Then protein kinase A also has a function. It turns phosphorylase kinase active. Then phosphorylase kinase turns glycogen phosphorylase active. Finally, glycogen phosphorylase can break down glycogen into glucose, and then glucose can be transported outside of the cell. So all of these signals work in conjunction, turning other signals off, turning other signals on in order to maintain its function. So how are these signals regulated? So signaling molecules may be turned on, turned off, depending on any hydrolysis. So for example, or any phosphatases, etc. So for example, as we mentioned with phosphates, you could have inactive enzymes. ATP protein kinase binds a phosphate to the enzyme. Once that phosphate is bound, it could be active. Then phosphatase takes off that phosphate and then returns it to its original state. For phosphate, though, the opposite can also be true. You could have an active enzyme. The active enzyme gets an ATP bound to it, then it becomes inactive. Then phosphatase takes off the phosphate, making it active again. So it could work both ways for ATP protein kinases. For GTPase, it's typically only one way. You have an inactive G protein, a receptor binds to it, activating the G protein coupled receptor. GDP becomes GTP. Now you have an active G protein. Then GTPase gets rid of the phosphate to return it to its original state. You always have to return everything to its original state in order to make sure equilibrium is maintained. And once again, lastly for ATP adenylyl cyclase, which is an effector protein, can be activated, turns it to CAMP. But then CAMP, you don't want multiple CAMP throughout the cell because as we saw before, CAMP turns off glycogen synthase. What if you need glycogen in your body? Well, phosphodiesterase can turn CAMP into AMP, allowing glycogen synthase to be turned on. And I believe that's everything. So if you guys missed the attendance form, please scan the QR code to fill out the attendance. Please scan the feedback form as well. It helps make sure we know what we need to improve on and also make sure that we can continue more review sessions for exam 2, exam 3, the final as well. If anyone has any general questions on how to study for the exam, how to do well for the exam, how to do well in the class in general, definitely now is your time to chat. So yeah,
Um, Hi. I have a question about how will I do well on the exam?
so for the
Yeah.
exam, what I recommend for most students to do is to
Mhm.
study from the slides specifically.
Okay.
The reason is Dr. Fath, Dr. Garofalo always mention their tests are taken always from the slides because the textbook itself has too much information that's not necessarily important for the curriculum.
Mhm.
So the textbook
Yeah.
should only be used as a guidance if you don't understand something on the slides. It shouldn't be your main form of study. So study from the slides specifically, because if you study from the slides, the thing is, Dr. Fat and Dr. Garofalo can test on anything from the slides, no matter how small it is, no matter how big it is. So study
Wow.
everything from the slides. That should be your main form of studying in order to do well for the test. And also
Wow.
with this test, the good thing is each chapter is typically split up evenly, and even within the exam itself, you're not gonna have a chapter 7 question at the beginning and then a chapter 2 question at the end. It's usually like the first 10 questions of chapter 2, next 10 chapter 3, next 10 chapter 4, etc., right?
Also basically like organized.
Yeah, it's
Wow.
pretty organized. But like I mentioned, it can be taken from any slide within that. So I definitely recommend that. Sorry, someone raised their hand.
Thank you so much.
Of course. Are the sample questions posted on Brightspace complicit to those on the actual exam? I'd say around like 80%. Typically, the sample question is a little bit easier, I'd say, than the actual exam. However, one thing, the actual exam itself actually has a similar structure in the sense that one thing you'll notice, Dr. Fath and Dr. Garofalo's questions are very direct. It's very, what is this? What is the function? If X doesn't work, what will happen to the function? For example, in the signal transduction pathway, if CAMP is turned off, what will happen, etc. These questions are very direct. The main issue that students typically face is obviously the amount of information that you're going to have to memorize throughout each chapter. But like I mentioned, the questions themselves are usually very direct. There typically won't be any like trick questions where there'll be like multiple answers. But once again, it is a lot of information to memorize. So I definitely recommend studying from the slides. Another thing that I did that I recommended to students and it worked well for them is recording lectures. The reason is some of you guys may notice that the slides themselves, sometimes they contain like lots of sentences. It explains it really well. But sometimes the slides are only diagrams where you need someone to explain what's actually going on in the diagram. So in those cases, like for example, even for my slides, I had diagrams where I explained. So for Dr. Fat and Dr. Garofalo, I definitely recommend recording lecture, which I know they're perfectly fine with. If you record lecture and then re-listen to it, then obviously you're going to be able to understand what they're talking about when it comes to these diagrams. And sometimes you also pick up on hints as well. Sometimes there are slides where Dr. Fat and Dr. Garofalo focus a lot of attention on. If they do, you know that that slide will be definitely on the exam. Sometimes there are slides where you see that it has a lot of information on the slide itself, but then they go through it quickly. So you know that that's probably not as important of a slide. So you're going to be able to pick up on like these little habits that they have. Yeah, they are straight to the point. What's the best way to concept of G proteins? The best way to remember the concept of G proteins, honestly, like I mentioned, with single transduction pathways, they're all very similar. So even with like the um phosphate uh phosphate receptors, they're very similar to G protein coupled receptors. So as long as you memorize each step individually, they're pretty similar for no matter what the secondary messenger or what the receptor is. Can you show the different bonds in water, like covalent and hydrogen bonds? Yeah, give me 1 second. I have to go all the way back. Uh, okay. So here, as I mentioned before, for hydrogen and covalent bonds, the main thing you need to know, hydrogen bonds occur between molecules. They don't occur within the molecule itself. So when we refer to hydrogen bonds, we typically say hydrogen forms the hydrogen bond with oxygen, nitrogen, and fluorine. However, it occurs between molecules itself. So two separate molecules. The hydrogen bound to the oxygen here is a covalent bond in and of itself because they're sharing electrons. But the hydrogen here bonding to the oxygen of a separate molecule actually forms the hydrogen bond. So yeah, is there a good technique to tackle multiple choice questions? So like I mentioned, because the questions themselves are pretty direct, there's always going to be a few answer choices that are like, they're definitely not the answer. So then it just comes down to your memorization, what you remember. One thing I remember for tackling multiple choice questions, sometimes the questions can be related to each other. So like I mentioned, because they're organized, question 2 could be related to question 8. So the answer for question 8 could give you the answer for question 2, or like the question itself couldn't state the answer for another question. So definitely keep that in mind because they're all related to one another. The timing on the test, would I say it's tight? I've seen students finish in 30 minutes. I've seen students take the whole time. It depends on obviously how much effort you put in to actually memorize all the information. Because it's very direct, there are students that do very well and they finish within half an hour because they know what the answer is for everything. However, there are some students where they want to take their time, look at each question, etc. It completely depends on you, but there typically isn't that many students that stay until the very end. It's usually only like 10 to 20 out of like a 100, 200, 300-person class, which isn't that bad, I'd say. The main reason students do poorly, I believe, it's that it's obviously a lot of information to memorize. And like I mentioned, because they can take information from any slide, you really do have to put in the effort to look at every slide and put in the effort to memorize every slide. Because of that, there are some students where, let's say one chapter is shorter than the other. They think, oh, I don't need to put that much information in understanding the information. However, what they fail to realize is that each chapter is weighted the same on the exam. So if they fail to study specifically for one chapter, then that's a whole section that they may not do that well on. So the main reason is just that it is a lot of information. So you guys have to find your own study methods in order to try and absorb all that information, take notes or re-listen to lectures, which is what I did, or even just attending these sessions, studying well in advance. There are lots of different ways, but you have to put in that effort to actually memorize everything. Are the adaptive quizzes helpful? So the adaptive quizzes are helpful in the sense that like it is extra questions. It helps you know more information, but it's not, I don't think it's necessarily pertinent to what's actually on the exam itself. Dr. Fat typically doesn't use those questions. And even then, sometimes those questions can also be extremely specific because it's dependent on the specific chapter. If that or whatever section they're talking about, if that quiz has extreme focus on something super specific that Dr. Fath didn't really talk about that much, then it's not going to be that helpful. So I wouldn't recommend studying using Achieve quizzes. I'd actually just focus purely on the slides. Textbook and Achieve quizzes should be a secondary material if there's something you don't understand. Of course, are there graphs, pictures, and models to analyze? Yes, there are. Sometimes you're going to have, like, for example, I mentioned for the cells, there could be a picture of a cell with part of the organelles missing, and you're going to have to say what organelle is which. And then there could be a question, a name organelle A. Or it's going to be multiple choice, but what is organelle A? What is organelle A's function? If you don't know what organelle A is, then that's two questions that you're missing out on. So there's going to be graphs, pictures like that on the exam. Is it good to read the slides ahead of time before lecture? Of course. I definitely recommend that. That way you have a general idea of what you're getting into. Especially for the future chapters. Once again, exam 2, you're gonna get into cellular respiration, photosynthesis. It is a lot of material because there's lots of molecules that you need to memorize for each process. So I definitely recommend reading the slides ahead of time so you have a general basis of what's going on. I actually recommended people to, even if they don't have time to read the slides, I'd recommend watching like the Amoeba Sisters or any like YouTube videos on whatever chapter they're about. That way you have a better idea of what Dr. Fath is talking about. Because there are times where if you don't have that previous knowledge, you're going to go into class, Dr. Fath could be talking about specific molecules and you're not going to understand what that molecule is. So having that prior knowledge, I'd definitely say is helpful. Maybe even watching like a 10-minute video helps. And yeah, like Khadija said, practice exams are a great way to gauge the formatting of the questions. Not necessarily the difficulty, but at least the formatting and how Dr. Fath and Dr. Garofalo word their exams. Another general thing for the course as well, lab is 40% of your grade, homework is 10% of your grade. So lab plus homework is 50% of your grade. Obviously, laboratory instructors differ in terms of difficulty, how you perform in the class, etc. However, there are some students that typically, if they're not performing that well on exams, they have an opportunity to make that up on lab and homework. So don't disregard your lab and homework just for the exams, because biology is the only course where it's worth homework and lab is part of your actual grade, 50%. So even if you're not the greatest exam performer, if you know that you're better at writing lab reports and you make sure you do your homework on time, that can significantly boost your grade to help you pass, get a B, get an A, etc. So don't disregard one for the other. In future lectures, how do you recommend engaging in his class and understanding his talks? For me, like I said, I just recommend listening, recording lecture if you can, taking notes during the lecture. Try your best. Try your best. Sometimes, obviously, he goes fast through slides, so there may be certain slides you don't write all the information on, which is why take notes during the lecture, record the lecture as well, so that way later on when you're studying it, you already have some of the information down, and you can complete that information when you re-listen to it. So yeah, the general study tips, study from the slides. Don't disregard lab and homework. Make sure you focus well on both because you can definitely pick your grade up on both. And good luck for tomorrow, of course. If anyone has any extra questions, I'll be here. Khadijah's here. Of course. And hopefully you guys filled out that form, the feedback form, so that way I know what to improve on, and then we could also help you guys. Of course, of course. Good luck on tomorrow. Yeah, these slides will, oh yeah, another thing. These slides will be posted on the Queens College Learning Commons YouTube, or not the slides, the recording will be posted on the YouTube. And actually, I did an exam 2 and exam 3 review last semester as well. So if you guys want to get a head start on those slides, on what I'm going to talk about for exam 2 and exam 3, then I definitely recommend looking at the YouTube because I already have the reviews are already posted from previous years, and they typically should be about the same chapters, barring like one or two. So if you guys want a head start, I definitely recommend Queens College Learning Commons YouTube. I already have reviews for exam 2 and 3 up, and this should be posted by tonight as well. Have a good day. Yeah, so just search up Queens College Learning Commons on YouTube. You're gonna find the YouTube with a bunch of review sessions for CS, bio, etc. They're not, I don't think they're separated into playlists, but if you just scroll down, there's not that many videos. So if you just scroll down, you'll see biology exam 2 review session, and then exam 3 review session, something like that. You'll definitely find that.
Will I find that in like Brightspace?
Wait, sorry, 1 second. Yeah, it does say Bio 105. So it says Bio 105 exam review session number 2. And then sorry, what'd you say, Tejas? Of course.
Yeah. Will this go be on Brightspace or like?
The recording will be on YouTube. I don't think this will be on Brightspace itself, like the slides.
Got it. Thank you.
Mhm. Have a good day, everyone.
Okay. Bye. It was nice to see you.
Yeah,
Have a
of
great
course.
day.
Yeah, so no, the slides won't be posted to Brightspace,
Mhm.
because Brightspace, Dr. Fath obviously wants it to be what he's teaching, his slides, etc. But the recording will be posted on YouTube for you to watch.
Excellent. Thank you so much.
Have a great day, everyone.